Okay, so let's talk about chapter 6. So chapter 6 is muscle. So this whole exam unit is bone, muscle, and the nervous system, which is a pretty daunting unit, but we're going to try to get through it together. So muscle tissue is one of the 4 primary types of tissues. We talked about skeletal, cardiac, and smooth muscle a little bit, specialized for contraction. So one of the 4 types of tissues, again, is muscle, connective, nervous, and epithelial tissue. So we're going to talk about the skeletal system, or the muscular system, basically, here, by talking about muscle tissue and the 3 different types. Skeletal muscle, again, moves the body by pulling on bones. So we have an example of skeletal muscle here. This is cardiac muscle. It's only found in the heart, and that cardiac muscle is utilized to pump blood. And then smooth muscle you can find in like the walls of blood vessels, and the walls of the digestive system organs like the small and large intestine, the stomach. That smooth muscle basically moves fluids and solids along the digestive tract and can also constrict and dilate blood vessels. So skeletal muscle tissue functions basically by moving around. So flex and extend your elbow, or shake your head no or yes. That is your skeletal muscle contracting and pulling on tendons that are attached to bone to move them. We just talked and finished the skeletal system. So we've talked about tendons a little bit before, and how some muscles connect to bone by way of its tendon, and the bone markings on those bones where tendons connect. But skeletal muscle can also maintain body temperature and body posture and position. So there's some muscles that are kind of constantly active that maintain posture. So how many of you just kind of sat up straight and straightened your spine and were slouching over? So that's body posture and position that is maintained by skeletal muscle. Supporting soft tissues. So tighten up your abs for me real quick. The abdominal muscles create a wall in the floor of the pelvic cavity as well that hold in your digestive system organs and organs in the pelvic cavity as well, and protects them. Some muscles, skeletal muscles, guard entrances and exits. So there are circular muscles that basically reside or are located at the very end of like the digestive tract or the urinary tract. And they form these structures called sphincters. It's just a circular muscle that can constrict and dilate, kind of close and open to allow or not allow certain foods or substances or liquids to get through that area. So like to basically control swallowing and defecation and urination. So if you've ever had to run to the bathroom to pee and you've had to hold it, you can kind of squeeze a sphincter and hold it while you're running to the bathroom. That is an example of a sphincter that you have control over. It's skeletal muscle. I've mentioned maintaining body temperature. Basically, muscle contraction requires energy, and that produces heat that can keep your body temperature regulated. And also, it's a nutrient reserve. There is some form of blood sugar, which is glucose storage. There's a storage molecule that's called glycogen. So you can store and even produce glucose in a muscle and store it there as well. The structure of a skeletal muscle. So think about a bicep muscle that we've mentioned before, or one of your abdominal muscles. There's a gross structure that is surrounded by connective tissue layers. And there's 3 connective tissue layers that surround different components of the muscle. The epimysium, and remember epi is on top of, right? It surrounds the entire muscle. It's a dense layer of connective tissue. It is dense, irregular connective tissue, something we talked about in epithelial tissue, or in tissues way back when. The perimysium surrounds and basically creates what's called a fascicle. I'll show you in the next slide. And the endomysium surrounds an individual muscle fiber, which is just a muscle cell. Tendons are bundles of fibers that are basically created from dense regular connective tissue that attach skeletal muscle to bone. those collagen fibers in the connective, there are collagen fibers in the connective tissue, those layers, the endomysium, the epimysium, and paramysium. There's some of the collagen fibers extend beyond the muscle at the end of it into and bundle into fibers that become the tendon, which is dense regular connective tissue. And then there's some muscles that have what's called an aponeurosis, which is basically a broad sheet, a tendinous sheet, as opposed to like you think a tendon is kind of like a tubular kind of, long, thick structure. An aponeurosis is like a sheet of paper. It's flat, but it's also a point of attachment for muscles similar to a tendon. And those on the other end, from where muscle and tendons connect, the tendons connect and they're collagen fibers from the tendons that basically work their way into the matrix of the bone through the periosteum, which is something we talked about in the last chapter. So here's a figure of it. There is the tendon here, bunch of stuff pointed out here. Here's a whole muscle. So the epimysium, epi is again on top of, myo is muscle. So the epimysium is this connective tissue layer around the entire muscle. I think this is a forearm muscle. The epimysium continues, some of the collagen fibers extend into the tendon. And if you take one of these smaller circular structures here and blow it up, that's called a fascicle. So there's a bunch of muscle cells that are grouped into what's called a fascicle. And that fascicle is surrounded by the paramysium, more connective tissue. And then one of these individual muscle fibers is here surrounded by the endomysium. So each one of these, this whole muscle fiber here, this is the paramysium. There's two areas where this is shown, paramysium. And then the individual muscle fiber is here. And then if you basically pull that out, there's the endomysium, a thin layer of connective tissue that surrounds that. So muscle fibers are the same thing as muscle cells. They contain, skeletal muscle fibers contain potentially hundreds of nuclei. There's a bunch of really small cells called myoblasts that fuse together to form one functional skeletal muscle cell or fiber. And that's where they take basically all the nuclei from those myoblasts, fuse together with their membranes, create a muscle cell with bunches of nuclei. And they are filled with small structures, small cylindrical structures called myofibrils. And myofibrils are made up of even smaller structures called myofilaments. And there's two types of myofilaments. One's called the thick filament, one's called the thin filament. So here's a look at that. So here's a muscle cell. And this muscle cell and skeletal muscle is striated, right? So I'll talk about why it's striated here shortly. These little dots here are myofibrils. So if you pull those out, remember myofibrils are smaller structures that pack inside a muscle cell. So here's a myofibril. This long myofibril is blown up here. And each one of these there's dark purple lines here and then red lines there. Those are myofilaments. So this is one long tubular myofibril. It's made up of myofilaments. Myofibrils pack inside one muscle cell, muscle fiber. And that muscle fiber, that muscle cell is what contracts in a muscle. The sarcomere is basically, so myofilaments, the thick filament and the thin filament, kind of group together to form a functional unit of skeletal muscle called a sarcomere. And at each end of a sarcomere, one end and the other, is a Z-line. That's the boundary between two adjacent sarcomeres. And basically they line up end to end in series. So one sarcomere and the next share a Z-line, and then that sarcomere shares a Z-line with the next sarcomere and the next and the next. And they line up in series, kind of in this long row, to create a myofibril. At the center of the sarcomere is an M-line. And those are the two most important structures in a sarcomere. So let's take a look at it. So right here is a Z-line. It's also called a Z-disc. Here's another Z-disc or Z-line. From one Z-line to another Z-line is one sarcomere. This is the contractile unit of skeletal muscle. So when muscles contract, like when you flex your arm and your bicep contracts, the tiniest, there's millions of sarcomeres that are all shortening so that this Z line is moving towards this M line, and this Z line is moving towards this M line, and the whole sarcomere collectively shortens. So this M line is another important structure here. It's the middle, the M line, the middle of the sarcomere. And when sarcomeres shorten, when the muscle contracts, the Z lines move towards the M line. And every sarcomere has 2 Z lines and 1 M line. So when the Z lines move towards the M line, the sarcomere shortens and the muscle contracts. One sarcomere is made up of a bunch of thick and thin filaments, and the thin filaments attach to Z lines. So remember, the Z lines are on each end of the sarcomere. So let's flip back to this. This Z line, right here, this pink line, these lines are thin filaments. So there's thin filaments here anchored to that Z line, and thin filaments here anchored to this Z line. Okay, so we've got now one of the 2 myofilaments, the thin filament. It has 3 proteins that make up the thin filament: actin, tropomyosin, and troponin. So I'm going to flip to the next slide and show you this and talk about what's on the previous slide so that we can figure out what the heck's going on here. So here's a Z line, and there's my thin filament connected to that Z line. If you blow that up, this is what a thin filament actually looks like. So actin is the major contractile protein on the thin filament. So actin and myosin are the big contractile proteins. Actin is this red and yellow protein here. And that yellow part is a binding site. We'll talk about what that binding site does later. Well, tropomyosin is this kind of bluish green filament that winds its way around actin. And as it winds its way around actin, it actually covers, in a resting muscle, it covers those binding sites on actin. So you've got actin, the contractile protein that is involved in muscle contraction. But tropomyosin covers the binding sites on actin when muscle is relaxed. And then the last protein is called troponin. And troponin has a binding site for calcium, which is really important, and we'll talk about why later. So for right now, you should know that calcium binds to troponin when muscle contraction starts. And then troponin is also bound to tropomyosin. So lots of T words going on here. The thick filament's mostly made up of just myosin. And myosin has tails. A myosin protein has tails, and it has a head. And the head of the myosin is what binds to actin on that binding site and creates a connection or a linkage between the 2. So basically that myosin head can swivel. It has an elastic component where it can bind to a binding site, and basically as the head swivels, it pulls, and then it will release. So it can bind to actin, pull on it, release from actin, and then re-energize, so it can recoil after it stretches. It has this really cool feature. So this myosin head, so there's a little tiny myosin head. This is the M line, and it's all of the thick filament. And this is just one myosin molecule. It has 2 heads on it. So each one of these myosin filaments collectively contributes to the entire myosin thick filament. And here's the hinge region. So the myosin head can swivel and pivot, basically, and pull. So the head of the myosin would bind to actin, grab a hold of it, swivel and pull on it, and then release it and swivel back to its original position. So the myosin head can kind of stretch and recoil over and over and over again. A couple of other structures that are really important in skeletal muscle that are not part of the sarcomere, but work to help the sarcomere do its job, basically. One of these structures is called the sarcoplasmic reticulum. It's an enlarged section. It's basically an organelle inside the muscle cell. And remember, sarco means muscle. The sarcoplasm is the cytoplasm of the muscle cell. The sarcolemma is the muscle cell membrane. So the sarcoplasmic reticulum is an organelle inside the muscle, inside skeletal muscle, that holds calcium. It's a storage depot for calcium, and calcium is important in initiating muscle contraction. Without calcium, muscles won't contract. So, and it's on either side of what's called a T-tubule. So on here, let's talk about what a T-tubule is. So again, I'll mention, as I did on the last slide, the sarcolemma is the muscle cell membrane. So the sarcolemma is just the membrane of a skeletal muscle cell or any muscle cell. And it has a membrane potential. We'll talk about what that does later. But basically, there are infolds. So that the muscle cell membrane folds in on itself to create almost the look of a T. And these T tubules are called transverse tubules, are basically infolds of the cell membrane. So they're continuous with the sarcolemma. They are part of the cell membrane. It's a part of the cell membrane that folds in on itself and creates a right angle. And that allows a passageway from the surface of the cell down into the muscle cell so that the electrical signal that's making its way through the cell can get down into the deeper parts of the cell. We'll talk about why that's important later. So hang on to that for right now. So here's a couple of structures. There's the sarcolemma, the muscle cell membrane. That sarcolemma folds in on itself. So there's a little opening right here, but follow that opening down in. So right here is a T-tubule as it makes its way down into the innermost components of the muscle. So underneath here, look, this is a myofibril. So these T-tubules are surrounding myofibrils. So there's the thin filament underneath there and the thick filament, right? You can see an M line right there, right? So the position of the M line is there. So you've got the infold of the cell membrane, the T-tubule surrounding the myofibril. And then this blue thing here is the sarcoplasmic reticulum where calcium is stored, okay? So take a look at this. You have a T-tubule that folds around, and then sarcoplasmic reticulum where calcium is located. So the close connection between a T-tubule and the sarcoplasmic reticulum is really important. And we'll come back and talk about why here shortly. Actually, right now. So the nervous signal or the action potential from a motor neuron. So in skeletal muscle, we're going to talk about how skeletal muscle fibers contract. And in skeletal muscle, you have to have a nerve that's communicating to the skeletal muscle to get it to contract. So the initial signal for muscle contraction is coming from a nerve. It's coming from the nerve from the central nervous system to the peripheral nervous system. So this nerve basically joins or junctions with the muscle. It's what's called a neuromuscular junction, right? So neuromuscular, nerve and muscle. The junction between the nerve and the muscle, that's what that's called, the neuromuscular junction. The part of the nerve that creates the neuromuscular junction is called the axon terminal. The part of the muscle cell membrane that creates the other part of the neuromuscular junction is called the motor end plate. So you've got the axon terminal and the motor end plate. This is nerve, axon terminal, motor end plate, the sarcolemma. So the part of the component of the sarcolemma that makes up one part of the neuromuscular junction is called the motor end plate. Okay, so follow me with this. We've got an action potential that has traveled, this electrical signal that has traveled all the way to the end of a neuron and reached the axon terminal. So right here is this red lightning bolt where the action potential has reached the axon terminal. It's really important, okay? Acetylcholine is an example of a neurotransmitter. So acetylcholine is actually held inside these vesicles. So this is a membrane-bound vesicle. Look, the membrane here is blue, and the membrane here is blue. So the membranes from here to here are the same. That's important. So this membrane in this vesicle here contains little red dots. Those little red dots are a neurotransmitter or acetylcholine. So the job of the action potential, once it makes its way all the way to this axon terminal, is to stimulate the release of neurotransmitter, acetylcholine, right? There's an enzyme there that helps break it down. It's called acetylcholine esterase that breaks down acetylcholine, but that's in the synaptic cleft. We'll come back and talk about that later. So as the action potential, basically the action potential reaching this axon terminal is the stimulus to release neurotransmitter into this space here, into the neuromuscular junction, right, between the nerve and the muscle. That's what these two slides are just mentioning, right? So the action potential, its job is to stimulate the release of acetylcholine. Acetylcholine then has a receptor on the motor end plate. And that receptor, when acetylcholine binds to it, it changes the permeability, how much sodium is allowed to move across that membrane. So acetylcholine diffuses across this space. That space is called a synaptic cleft. So basically, once acetylcholine is released from the axon terminal, it diffuses into this space. That space is called a synaptic cleft. And across there, it binds to receptors on the sarcolemma, the motor end plate here. And what that does is that receptor is a signal. When acetylcholine binds to it, it's a signal to change the permeability of the membrane on the sarcolemma's membrane to sodium. And when it does that, basically sodium ions rush into the cell. And when sodium ions rush into the cell, another action potential, another electrical signal is generated on the sarcolemma. So right in this motor end plate, you see all that red there? That's the designation that a new action potential is being generated. So to summarize, the first action potential in the neuron stimulates the release of acetylcholine from the axon terminal to send it into the synaptic cleft. As it crosses the synaptic cleft, acetylcholine binds to its receptor on the motor end plate. And the motor end plate is part of the sarcolemma. When it binds to that receptor, it activates the receptor and changes the membrane's permeability to sodium and allows sodium to rush into the cell. And by doing that, that sodium rushing in actually stimulates the generation of another action potential. But this action potential is now on the sarcolemma at the motor end plate. When that action potential is generated here at the motor end plate, so follow me to here, right here is the neuromuscular junction. That action potential is generated here, but then spreads out through every which portion that it can of the skeletal muscle cell. So one skeletal muscle cell has one neuromuscular junction. And at that neuromuscular junction, an action potential is generated on the sarcolemma and then spreads out across the sarcolemma of the entire muscle cell. And then it spreads down into the T tubules, right? So now the action potential is spreading across the entire sarcolemma, and then it spreads down into a T tubule, and that T tubule meets up with the sarcoplasmic reticulum. And I said I'd come back to why that's important. So this T tubule, the action potential that propagates down this T tubule stimulates the release of calcium from the sarcoplasmic reticulum, which is why their close association is so important between the T tubule and the sarcoplasmic reticulum. Okay, that was a lot. We're going to continue on. So if you need to, go back and replay parts of that video or parts of the previous part of this video, and make sure that you understand muscle, how the neuron, how it releases acetylcholine by way of the action potential, what that acetylcholine does, binds to a receptor and changes the permeability of the membrane to sodium to generate another action potential. And then that action potential spreads across the muscle cell membrane, the sarcolemma, and down into T tubules, where it meets the sarcoplasmic reticulum and stimulates it to release calcium. From this point forward, the release of calcium is what takes this signal that's been given to it and initiates muscle contraction. So we're going to go through a couple more steps here. At rest, when a muscle is relaxed, the myosin head is energized and it's actually pointing away from the M line. So we're going to come back to the sarcomere here and talk about how, once calcium is available, how myosin and actin actually interact and cause muscle contraction. So basically, ATP is used to provide energy to myosin so that that head can swivel and basically stretch and recoil and stretch and recoil. So right here, this is a figure that I couldn't get to blow up very easily. So if you have the book or if you have this figure and you can blow it up so it is clear, go ahead and do that because I want you to see the close components here. So right here is the green ball, that's calcium. Calcium has now been released from the sarcoplasmic reticulum based on all the steps that we just talked about. So calcium arrives in this area where there is both myosin heads and actin. Okay, so the thin filament is here. Remember these balls, these red and yellow balls were actin. The blue is tropomyosin. And then there's little brown molecules called troponin, right? Remember, troponin has a binding site for calcium, which is so important. So when calcium gets released from the sarcoplasmic reticulum, it binds to troponin, right? So calcium here is binding to troponin, and that's the signal to start muscle contraction. Troponin changes shape when calcium binds to it. And as it changes shape, it pulls on tropomyosin. And remember, in a resting muscle, tropomyosin is covering the actin binding sites where myosin can bind. So in a resting muscle, myosin cannot bind to actin because tropomyosin is covering it. So calcium then binds to troponin, which is bound to tropomyosin, and moves tropomyosin out of the way of the actin binding site. And if tropomyosin is now out of the way of the actin binding site, myosin can bind. And it does. Myosin heads bind to actin, and the binding of myosin to actin is that formation is called forming a cross bridge. So when myosin binds to actin, a cross bridge has formed, right? And then once the cross bridge is formed, the myosin head goes from this kind of energized, cocked back position away from the M line. So if the M line is over here on the left-hand side of the slide, the myosin head swivels from where it was energized this direction, and it swivels this way and pulls. You can see the arrow here, it pulls on the thin filament and basically pulls the thin filament that's anchored to the Z line towards the M line. Remember, ATP fuels all of this. So myosin head swivels, and then a new ATP comes in here. Myosin and actin detach from one another, and then ATP donates its energy to swivel the myosin head back into its original position. So this whole thing can start over again. Myosin binds to actin once calcium is bound and moved tropomyosin. Myosin head swivels, it pivots, and pulls on the thin filament, moving the Z line towards the M line. And then a new ATP molecule comes in, releases myosin from actin, and that cycle starts all over again. So this is called cross-bridge cycling. So where a cross-bridge forms, myosin binds to actin, and then it performs a power stroke. It swivels and pulls on the thin filament, and then releases. The cross-bridge detaches. So you should understand that whole cycle. Again, the free myosin head splits a new ATP, and it energizes it, and this whole process starts all over again. And that's basically the summary of a sliding filament theory, is that the thin filaments slide past the thick filaments, and the Z lines move closer together, something I've mentioned a couple of times. So the sliding filament theory just basically states that this M line stays right where it's at. And as myosin heads, these little dots here, swivel and pull on actin in the thin filament, these Z lines move closer together. The filaments, myofilaments, are the thin filament and the thick filament. And the sliding filament theory states that the thin filament slides past the thick filament as the thick filament pulls on it, and Z lines move towards M lines. Overall, when this happens, the sarcomere shortens. So the length of the sarcomere decreases. You can see that here. So I mentioned this is a myofibril. Okay, this whole structure is a myofibril. In a resting muscle, it may look like this. So there's one Z line, an M line, and a Z line, which makes up one whole sarcomere, and then another sarcomere, and another sarcomere. So all these sarcomeres line up end to end. And when one sarcomere shortens, they all kind of shorten together collectively to shorten the whole muscle. So here's a resting myofibril and a contracted myofibril made up of sarcomeres. These sarcomeres are made up of myofilaments, the thick and the thin filaments. And when that happens, when all this happens and muscle contracts, the tension that the muscle contracting basically is transmitted to bone. Remember, muscles attach to bone to move bone. So myofilaments basically attach to the very end of the muscle fiber, and then the very end of the muscle fiber attaches to tendons, and then tendons attach to bone. So there's a big series of connections here, and the result is when a muscle contracts, the bones move. So flex your elbow. When you flex your elbow, your bicep muscle is shortening. It's thousands and thousands and thousands of sarcomeres all shortening at once to contract the bicep that then pulls on the tendon, and the tendon's attached to the bones in your lower arm, the radius and the ulna. So it's actually the radius. Well, depending on what muscle you're talking about. So when the bicep muscle pulls on that bone in your lower arm, the radius, your elbow flexes. There's another muscle we'll talk about later that also attaches to the ulna. But collectively, when you flex your elbow, the muscles that flex your elbow are pulling on your ulna and your radius by way of the tendon. When a muscle returns to its original length, it's basically just a passive process. Contraction is completely active. You use ATP as a power source. The muscle contracts by using ATP that fuels the myosin head swivel. When your muscles relax, it's basically just gravity helps pull them down, the contraction of the opposite muscle. So an opposing muscle, like an opposing muscle to the bicep, would be your tricep. If the bicep flexes your elbow, the tricep extends it. So sometimes opposing muscles can help relaxation of a muscle. And there's some elasticity in the tissues that are stretched by contraction, and then they help that elasticity helps return them to the resting length. So it's a completely passive process. And I keep using this bicep as an example. So shortening of the bicep, contraction, lengthening of the bicep, relaxation. But here's the tricep that when it's contracting, it helps the bicep relax. When the contraction cycle ends, there's a couple of really important events that occur. The stimulus from the motor neuron, so that release of acetylcholine stops. And acetylcholine gets broken down by acetylcholinesterase, that enzyme that breaks down acetylcholine in the space in the neuromuscular junction, that synaptic cleft, which means the action potential on the muscle cell membrane, the sarcolemma, stops. And the sarcoplasmic reticulum, the storage depot for calcium, actually has a pump on it that can pump the calcium that was once released back up into it. And if calcium is being pumped back into the sarcoplasmic reticulum, it's no longer available to initiate contraction. So the muscle starts to relax. And when that happens and calcium is being removed and sent back up into the sarcoplasmic reticulum, it's released from troponin, and then troponin rolls basically tropomyosin back over the actin binding sites and covers them once again to be a resting muscle. All right, so I felt like that was quick, but it is what it is. So that's chapter 6A. We have 2 more lectures for chapter 6, B and C, so I will see you soon.